So hello everyone out there in BB2903 land. This is Dr. Buckholt coming to you not quite live from my computer at the moment. I'll try my best not to sneeze, cough, or drink into the microphone as much as I can avoid, but my mouth gets dry talking for 15 minutes. So I'll try not to make too many inadvertent noises. So sorry for those of you who don't know who I am, you won't see me until next week. So what I'll be giving you today is the sort of background lecture for the lab in microscopy. My co-instructor, Ali Hunter, will be doing a separate lecture, which you should all make sure you listen to, on how the sort of class will work, how the sort of clerical things will go. And for all of you, even if you've taken a 2900 lab before, definitely pay attention, because we're going to start a new scheme on how we're going to write lab reports, which hopefully will do a better job of teaching you the best way to write one, and hopefully be a little bit less work for both you and the TAs, particularly because there are 100 of you in class. So make sure you pay attention and watch that lecture also. As I said, I'm Dr. Buckholt. You don't ever have to call me that again. If you need to, that's fine. Probably professor, but Mike is more than sufficient, and I'm sure Ali will tell you the same thing. So, on to background for microscopy. The first couple of slides you'll see here are things we're actually not going to cover. They're things we would normally cover if we had a lecture, but there's the background of what Ali's going to cover in the other lecture, but they'll give you an idea of sort of how things work. So there's a lot of class stuff. Ali will cover all that. The cool thing to remember is that we have videos on how to do everything now. I don't know if she may or may not tell you, but we're going to give you a slightly different way to access the videos than we have before. For those of you familiar with 2900, we're going to have everything contained in a Wiki site. So you'll need to go to the Wiki from the pre-lab to see the videos ahead of time, and as usual, the links will also be in the lab procedure. So when you get to pre-lab, there'll be a link to go directly to the Wiki so that you can find it and you can bookmark that or use it however you want. So that's that, and Ali will go over the real details of this. And a lot of you know we collect data in class using the clickers. We're actually going to collect data in class this term, hopefully in a slightly easier fashion by using Google Docs, and we'll talk more about that when you guys actually come to lab. Again, Ali will hopefully remind you all this. I'll remind you once again, the more often the better. Remember proper lab attire, which isn't so hard in the winter time. Completely covered up, closed-toed shoes, and make sure you bring safety glasses. We do have loaners if you need them, but you never know who wore the safety glasses before, so it's always nice to bring your own. All right, so that's just a few words in the background that Ali will cover in the other lecture. And what we'll do now is sort of move on to the sort of background. The course objectives, Ali will again hopefully cover. If we don't, it's in the syllabus. What we want to do is move on to actually talking about what we're going to practice this week is the introduction to microscopy. And we want to do this because no matter who you are, if you're going to continue in anything related to biological science, you're probably going to have to use a microscope for something. So now is as good a time as ever to start learning how it works, how to play with it, how to get the best view out of it. So this whole first lab is designed to just play with the microscope and look at different kinds of things under the microscope and get an idea how it works. And we'll be using them for the rest of the term. So, now's a good time to learn how to find everything, how different parts work. So one of the things we'll do is learn to identify, describe different parts of the microscope, what they're actually for. Hopefully by the time we're done, you'll be able to use the microscope more or less properly, learn how to care for it, which mostly means cleaning the lenses appropriately. And in this case, we're going to be using, at some point, slides with an oil lens. And in which case, this mostly means not getting the oil on either lens, and if you do, making sure you get all the oil cleaned off of everything. You're going to prepare a few wet mount slides, so you learn how to do that. You're going to look at slides that are both stained and unstained, so you can get an idea of what stain will do for you and why you might want to do it. And lastly, the other thing, you'll find that 4 or 5 times during the term we're going to collect data from the whole class and do some statistics on it, so you can learn how to do some really basic statistics and get an idea of why it is in science you probably want to have more data than just your one time around or just your own to draw conclusions from. So this first time around we're going to do a really simple experiment looking at yeast budding, which will help in your microscopy skills because yeast are really hard to look at. And we'll do a simple mean standard deviation analysis for that as far as stats goes. So since we're talking about microscopes, there are essentially 2 different overall kinds of microscopes, and then there are other ones, specific types that get divided under those. But the 2 most common types are the simple and a compound microscope. So if I had all of you here in front of me, what I'd be asking now is, can you think of an example of both of these? And more than likely, you would all be able to say, oh yeah, I mean, what you think of as a normal microscope is a compound microscope. What you may not always think of is, though, that simple microscope is, and now you can see my poor drawing skills and or writing skills, and what happens when you're running multiple programs on your computer at once. We'll see how this goes. Now, if any of you can actually read that, that would be amazing. It's actually a 2-word answer. And the answer is magnifying glass. So good luck if you can read my writing, because it's bad normally, and when you're trying to write with a mouse and a flickering program, it doesn't look so good. So anyway, a simple microscope is a magnifying glass. And what it really means is a simple microscope is something that magnifies with only one lens. What makes a compound microscope is it has at least 2 lenses to do magnification. And that's what we all commonly think of as a microscope. So that's generally what we'll talk about as we go on. The next slide shows you sort of what the differences are when you think about that as far as inverting the image and what it does for your magnification. The thing to remember of simple versus compound is the magnification you get when you talk about a simple lens. You only have this one lens, so the magnification factor is whatever you get from just that one lens. All right. If you're talking about a compound microscope, you have 2 lenses. And what you get is the total magnification of those 2 is a multiplication factor of those. So if this one down here is 10x, and this one is 10x, then the total magnification is 100. To get the same thing in a simple microscope, this lens by itself has to be a 100x lens, which is a little harder to do and harder to get light through. So that's just to give you an idea of the differences, and this is basically how the microscopes you're going to use work, the compound ones. There's a couple of lenses in there that do all the magnification. And for those of you who know us by now, you know it wouldn't be a real background lecture if we didn't give you some amount of history. And in science, it's always good to keep in mind what came before that allow you to get to the point we have. So the next couple of slides are just some examples of the very first microscopes. And these 3 in particular are pretty important in history. This one by von Leeuwenhoek, and my Dutch is terrible, so who knows if that's really right. And is important because it allowed for some of the first observations of really small objects and it did it in sort of an amazing way, which we'll talk about in a minute. And the Jansens are possibly responsible, because no one is absolutely sure, for making the first microscope ever. And you sort of see how it goes historically. You know, these first two don't necessarily look like what you might imagine as a microscope. This one definitely not at all. You know, it's more like a big magnifying glass, which really it is, and we'll talk about. And this one looks more like a telescope than a microscope. And you see, as you get down toward 1670, the microscope that Robert Hooke used is starting to look a lot more like what we expect of a microscope. It has some of the same pieces, which we'll review after a while, and actually had an external light source to allow you to actually throw some light on the subjects. So those are the sort of first historical microscopes, and you know, things progress slowly over the years. But you see that, you know, by the 1850s, the microscope looks a lot like what we have now. The cool thing is then they were made out of, for instance, all brass, which is interesting. And, you know, by the 1930s, they look pretty much like what we have at the moment. And the real difference between this and what you're going to see in the lab and what you're going to use in the lab is this one only has one ocular. All of yours are going to have two oculars. And though some of you will find it difficult, now is a good time to learn to actually open both eyes while we're doing this microscopy lab and look through both oculars. This microscope down here, you know, though this is now a few years old picture-wise, this is pretty much what a real research microscope looks like, and it'll cost you upwards of $40,000 or $50,000 or more, because it has all kinds of interesting optics, probably do fluorescence, has an embedded camera with all kinds of controllers. But that's sort of the state-of-the-art kind of research microscope, and they go up from there. So let's just give you a little historical perspective to see, you know, how we've gotten to the point where you guys can have a student microscope that's way better than any of those ones that came before. And a few more details about the people who made those microscopes who we're looking at. So the Jansens, the one that looked like sort of a telescope, we think were probably made around 1595. No one's sure whether Zacharias or Hans, his father, made the thing. And it had two lenses and an extendable tube, which allowed you to essentially focus and change the magnification. And it only went essentially from 3x to 9x. So you can imagine that you couldn't see very much with that. And today's magnifying glasses, in general, are way better than that. All right. The Robert Hooke microscope, that was the one that first one that started to sort of look like microscopes you saw at the bottom, is important because Hooke published this thing called Micrographica, and you know, if your Latin's any good, it is what it sounds like. You know, essentially small graphic. And the thing that he's really famous for and has come down through history to us is he showed a number of small organisms, but he's most famous for discovery of cells in cork. So you know, he looked at, essentially did sections of cork, the bark from the cork tree, looked at it, and what he saw under there looked like cells. And I'll ask you in a minute, and if you were in front of me, why he called them that, and I give you a minute to think about that. It'll be on the next slide. And the difference between him and some of the others is that his microscope was actually made by someone else. So he did the work, but Christopher Koch was the one who actually made the microscope. Another famous microscopist in history essentially was Marcello Malpighi. And what was really cool about this is that for quite a long time, the man named Harvey had proposed that blood in your body actually went around in a circle. That it was a whole interconnection of blood vessels somehow, but no one could prove it because no one could see the capillaries because they were way too small. They knew blood went out through arteries, came back through veins, and no one could find the connection. So it was finally Malpighi who managed with the microscope to actually see the capillaries and prove that Harvey's blood circulation theory was correct. So that was pretty cool. And the sort of interesting thing in all this is, so most of those microscopes above were actually versions of compound microscope. The interesting thing was von Leeuwenhoek, who used a single lens microscope. So that funky little thing, the very first one I showed you, that didn't look anything like a microscope because all it really is a huge magnifying glass. Made reports to the Royal Society in London all the way back in 1673 describing protozoa, bacteria, sperm, red blood cells. And he called them at that point animalcules, essentially meaning small animals. So that's kind of just a brief history of the microscope and the men who sort of got us to this point. So those are the people whose shoulders you're standing on at the moment. And so now we're down to the point is, if you're all standing in front of me now or sitting in front of me now, I would say, so why did he call himself, right? And, those of you who think back in history, what he would have been familiar with was the monasteries and the little cells that monks used to live in, or at least sleep in. Excuse me. And he thought that when he looked at the cork, all these little holes looked like the tiny little rooms that the monks lived in, and they were called cells, so he called them cells. So you can see how that one naming event, what was that now, more than 300 years ago, we've carried all the way down through to this time, and we still call all those parts in our body cells. So I want to make a little mention of it because it's just really amazing, and it's so cool that he's able to do this. That, how did von Leeuwenhoek manage to do that? He had a lens, microscope that only had one lens. So that meant that one lens had to be really powerful, and you actually had to be able to see through it, which at that time was not easy to do. There was most lenses at the time were made by essentially smashing molten glass between pieces of wood and then hoping you got something that had a curvature that would actually magnify light. All of those had some kind of optical problem. They all had magnifications of 50x or less. And as I said, remember most of the other microscopes were compound microscopes, so that meant you usually use two lenses put together. But if you think about it, remember the magnification multiplies from between lenses. Well, the other thing that magnifies between lenses is the aberrations or the problems with the lens. So if you put two lenses together that are not very good, what you get are one really crappy image. Right? So if you have the advantage is if you can make one good lens, even at a high magnification, then you can actually really see something. So what von Leeuwenhoek managed to do was make a single lens that had sometimes as much as 200x magnification power, which means you can resolve things down to 2 microns. And in a little bit we'll talk about what the resolution actually means. And no one's quite sure how he made it, but instead of the molten glass between wood bit, at some point we think he actually was blowing or grinding the lenses. The sort of sad thing is that he had about 500 of them sold after he died because apparently his wife at that point needed the money, and now only 9 specimens remain of something that was a pretty amazing technological feat for the time. Remember, that was 400 years ago. All right, so I think we're down to the end of our real history lesson. So what we want to do is do a little bit of review about what you're going to see in the lab this week. And I will apologize now. This is a picture of the old microscopes. You guys get to use nearly brand new microscopes that we just bought. So they have all the same parts, but it's not going to look quite exactly like this. What you see starting at the top is number 1 is the ocular lenses. And as I said before, all the microscopes we're going to have are binocular microscopes. So they're meant for you to look through with both eyes and both eyes open so you get the best image. And it's good to start learning that now, because if you ever have to do a lot of microscopy, looking through one eye gets very tiring and definitely will give you a headache at some point. The thing to keep in mind is it is in the instructional um, details that it gave you if you go look at the how to use microscope. And I don't remember quite sure if it's in the video or not, but you'll see that the oculars, one of them is adjustable, and that's meant so that if your eyes aren't quite the same, like mine, my right eye is stronger than my left eye, that you can adjust the microscope to compensate for that. So what you do is, whichever ocular doesn't have the adjustment, and usually it's the left one, you close your right eye, focus the microscope with just your left eye open, then close your left eye and look down through the adjustable eyepiece with the right eye, and then you turn the adjustment until the microscope is in focus. And now what you should have is both eyes should be able to see the same image in the same focus, and you should have a lot fewer headaches and life should be a lot better. The other thing you'll see is that there'll be a head on the microscope, and that head can turn in both directions. This is the most common way to have it pointed this way. That way you don't smash your nose down here onto the arm of the microscope, and there's a little knob on the microscope that will allow you to loosen or tighten it so you can spin this around. You definitely will see the objective lenses on the turret, and I'll show you some details about that in a minute. There'll be an arm that holds the whole microscope. And then the other really important things to think about are there's a down here 14 and 15. See there here. This is the adjustment back and forth, left and right, to make the stage that holds your slide go back and forth. So you need to learn how to move that. Remember, they'll always move in the opposite direction you think under the microscope, so practice that some. And then 12 here is the coarse focus, so that's how you focus the actual microscope, and 13 is the fine focus. Other things you should learn to play with are, and they'll look a little different on the microscopes, is there's a condenser and iris here so that you can play with how much light you actually let through and where it's focused so you can get a better image. And then 8 is the actual light source. So the difference on the microscopes you guys get to use is we have LED light sources in the microscope, so that you have really white light, so it gives you a really good image. The thing you generally want to learn to do is there'll be sometimes an on-off switch and a sliding switch that will increase or decrease the illumination. And generally you want to have the illumination all the way up and use right here the diaphragm to change how much light actually comes through, and that gives you a better image. It's not quite so much true now with the LED lights that we have, but it's definitely a good habit to get into, to use the light all the way up and adjust the diaphragm. So those are the parts. The next slide, which is replicated in your lab procedure, just gives you the details on each one of those parts if you want to go through it and figure it out. So I just wanted to go through a few concepts that are really important in microscopy. And you'll hear us talk about these words all the time, and hopefully after you're done with this, they'll have a lot more meaning for you. We want to talk about the difference between resolution and magnification, a little bit about field of view, and then the importance of staining. Why do we stain things? What will it actually do for us? Right? So when you think about resolving power and magnification, the resolving power of a human eye is 0.1 millimeters. And before we go too far, what I want you to think about is resolving power essentially is the ability to say, if there are, for instance, 2 dots under the microscope, I'm able to see them as 2 dots, as opposed to, if I have a really great magnification, I can blow them up really big, but if I see them as one really big dot, then they're wonderfully magnified but not well resolved, right? So you want to have magnification, you want to be able to go from this, but then see still 2 big dots as opposed to 2 little dots, right? So resolution and resolving power is the ability to separate 2 things that are really close together and see them as 2, not make them one great big blurry magnified thing, which doesn't really help you very much. So it's better often to have more resolving power and less magnification. So because the resolving power of the human eye is about 1/10 of a millimeter, the smallest thing you should be able to see is 100 microns. Which means is that in theory, we're going to look at amoeba this week under the microscope, you should be able in theory to actually see an amoeba on your slide. As I say in theory, I've looked 1,000 times and I can't see it, but then again I have old eyes. So you guys should give it a try. See if you can see the amoeba on the microscope. The confounding problem there is that there's very little contrast between an amoeba and the surrounding slide and water. What you're trying to do is find a clear thing or an almost clear thing in a whole bunch of clear things. So it's not so much necessarily a matter of size as contrast. But give it a try. You might be able to see it. So what you should see is a good optical microscope. So the ones that we have, and they're pretty decent, can resolve about 0.2 microns or 200 nanometers. So the reason for that is visible light has a wavelength of 390 to 700 nanometers, so the wavelength that we're able to perceive. To see something, it has to be big enough essentially to block a wavelength of light, which means it needs to be at least half a wavelength in size to obstruct light. So the smallest thing we can see is essentially half of 390, so that's why we see about 200 nanometers. So that means essentially the smallest distance that we can resolve is about 200 nanometers, and that means we generally have no trouble seeing bacteria, things like mitochondria in the microscope, because mostly those are about 500 nanometers. I'll show you a little chart in a couple of minutes to give you an idea of what you can see in what kind of microscope. So another good concept to keep in mind, we're not going to really use it to measure things so much this week because we're not going to give you little micrometers to do it with. But one of the things you need to think about is field of view. And when we talk about field of view in general, what it means is whatever you can see in that round circle when you look under the microscope. But what you can do is if you know the diameter of that round circle, or essentially the size of it, it allows you to measure and estimate the size of objects. And you can actually measure the field of view by putting a really tiny ruler, essentially, underneath the microscope and calibrating the size. Now you can really only do that at low power because at high power, you're essentially looking right between the little gradations on the ruler. But if you do it at low power, then you can easily convert to high power if you use the formula C equals A times B over D, where A is your field of view, whatever you measured. B is whatever magnification that was at when you measured it. And D is the magnification of whatever field of view you changed to. So if you measured it at, for instance, 10x, and you move to 40x, then you see what that does formula-wise. The field of view, essentially, anything in there is going to be a quarter of what it was in the original field of magnification. So just keep that in mind. There's a direct application or direct link between what the magnification is and what the field of view is. So if you want to use it to measure things under the microscope, it's relatively easy to do. So keep in mind that essentially the way we're going to use it this week, though, is field of view is just going to be what you can see under the microscope at one time, and that will be important when we talk about counting the yeast that you're going to count for the budding experiment. All right. So this slide is up here to give you an example of why it is good to stain things, and you get an idea about that when you look at, for instance, the yeast this week, because yeast and the amoeba also don't have much color to them normally. So there's not much contrast between them and the surrounding solutions or anything else. And so it's hard to get a really good image. And a lot of things are like that in biology. You know, on TV, they're really happy to show you that everything in the world seems to be colored and stained, and all the solutions we work with are colored. You guys probably by now know that that's not really true. Right? Half the things we work with in biology you either can't see or don't have any color at all. So sometimes you need to stain. So if you imagine, you know, this is an example is some small thing, generally cells you're looking at the microscope. You know, these happen to be plant cells over here, or these which are actually paramecium. You can see them. You can get a good idea of what structures are in there. And this picture is actually way better than it would look like if you looked at it under the microscope. But if you were to actually stain them, then you get something that looks more like this. You know, this and this. And now you can really get an idea, you know, see the structures inside the paramecium, really get an idea of the chromosomes inside these cells, really see the cell wall well. So generally, staining is going to enhance the contrast, and depending on what stain you use, sometimes it's going to label specific parts. So it's going to allow you to see things under the microscope you might not have been able to see, or for sure, at least more easily than you could have before. The one drawback is that, unfortunately, it's not always true, but many of the stains that you end up using in biology will actually kill the tissue or the organism that you want to stain. So you have to be really careful what you stain with if you want to have a live sample after you're done. But at the very least, he might be dead, but it'll be pretty. All right, so I know this is a very busy slide, and I don't expect you to really memorize and understand any of it, but it just wanted to give an idea that, you know, at this point in your career it won't matter as much. But if you continue to use microscopes a lot, it's good to remember that there's a lot of information about how the lens will be used or what it will do for you if you look on the side of the lens. So this drawing is of a 60x objective lens, you know, so the ones that go down in the turret at the bottom of the microscope, the ones that actually are right next to the slide. And it will tell you a lot of things, you know, who made it, which isn't necessarily very important. But some other things that are important is, you know, what is the magnification? Right? In this case, it's 60x. And if you take microscopy, the lab module, it probably talks some about different corrections you have. So this one has a planar correction, or it's for a flat field. It tells you whether there's any aberration corrections in the lenses. And other important things are what's a numerical aperture. And those are important because all the things on a microscope need to match to get the best image. So you need to know those things. And the two things that are most important for you if you just walk up to a microscope, maybe three, is this case, you sort of learned that most lenses 60x and above are actually oil lenses. But you won't necessarily know that unless you look at it. So you see here where it says oil. That means to get this lens to work right, what you have to do is put a drop of oil on the slide and actually turn the lens into the oil so it touches the oil. So you get a direct line from the slide through the oil to the lens. And that's because to get really high magnification, if you think about it, there's a lot of refraction that goes on. The light gets bent as it goes out of the slide, you know, through those surfaces and back into the lens. So to prevent that, what you do is essentially have a direct oil bridge so the light gets a lot better path into the lens. What it does mean is if you try to use the oil lens without having the oil on there, is you might not be able to focus it right. So if you're having trouble focusing, one of the things to look at is, do I have the oil lens turned down and not have oil on it? Right. So because the lens is essentially ground so that it's expecting the oil to be there. So it's one thing to keep in mind. If you can't quite focus it, that could be a reason. Two other really important things are, you'll see that it will actually tell you how thick the cover slip you use can be. And that's important is because this working distance over here, needs to be higher than the thickness of the cover slip that you use. If the cover slip is too thick or is thicker than the working distance, then your lens is never going to focus, because the focal distance of the lens is not going to be on the specimen on the slide. It's actually going to be inside the cover slip. So you're never going to see what you want. So always make sure that the cover slip thickness is less than the working distance thickness. So it's just one more thing, is if you say, I can't see or I can't focus, check that out. And quite often what you'll find is with the cover slips we have, they stick together and you'll have two. And if you have two of them, then they're definitely too thick for the working distance and it really won't work right. The last thing to remember is most of the lenses we have are spring-loaded on the end, so they will move generally out of the way. If, for instance, you're a little too close when you get to the, or too close to the slide. But in general, what you want to remember is that, and I don't think that I talked about it anywhere else before, is these microscopes are what's called, and we'll try the drawing thing again, and it is in the procedure, or parafocal. And that means that once you have the microscope focused on one of the objective lenses, when you turn to another one, it will be nearly, if not completely, in focus. And so as long as you do like we tell you and focus with the lowest power magnification first and really get it focused, as long as that you really have it focused, when you turn to the higher power magnification, even though you think it might be going to hit the slide, it won't, because if it was in focus, the next one will be in focus and will work. If the lens does hit the slide, then it meant you weren't really focused on the right thing on the previous lens, and we need to go back and look at that. Right? So if you have trouble with that, flag us down in lab, and we'll work with you until you can figure out what the real focal plane is and all that. But keep in mind they are parafocal, so once you have it focused, don't move the slide up and down, don't touch the focus, just flip the turn around, and then you should have very little work to do to focus it on the next lens. And that will be really important for the yeast, because it's really hard to find yeast on a high power lens if you haven't found them already on the low power lens. Alright, so this slide is just a little example of what you can see with what. Remember the human eye, remember we said could get down to about maybe 0.1 millimeters. So you can see a lot of eukaryotes with the naked eye, and that's really, amoeba, things that are a little bigger than that. When you get down into prokaryotes, things like bacteria, there are possibly a few that are big enough to see by eye, but not very many, so most of them are going to require the light microscope. There are a few viruses that are just big enough that you can see in the light microscope, but not many. And then everything else after that, most viruses, all the way down to proteins and atoms, you need to have some version of electron microscope to actually see. So this will just give you an idea of what you might need and what varying sizes of things you're going to be able to see depending on what you have available. So all of these again are in the how to use the microscope document, and somewhere in the procedure. I just want to go over a few to make sure that you are really careful. The way the lab's set up now, you won't have to carry the microscope very far. It should be in the bench, the cabinet right underneath your bench, so all you have to do is pick it up and put it on the bench. But as a reminder, like I just said, the things you want to do is always start on the lowest power objective. And the reason for that is that's the one that's most easily focused, especially if you haven't practiced very much. So learn to focus on with the low power objective in place. You want to have the condenser most of the way up and the diaphragm open, and use the diaphragm as much as you can to adjust the light as opposed to adjusting the light level with the power switch. The thing you shouldn't do is plug the microscope on with the power turned on, because you don't want to give full power to the lights right away, because that can be a little damaging. It's probably less damaging now that we have the LED lights, but we still want to be careful. So your microscope may or may not need cleaned. If you're not sure how, have us come over. Things you definitely want to do is use lens paper and make sure it's lens paper and not Kim's wipes, because Kim wipes can be a little bit abrasive and we don't want to scratch the lenses. And you should have lens paper either on a box on your bench or in the little skinny drawer right in front of you. So make sure you use lens paper and lens cleaner to clean them off, especially if you got oil on the lens. You make sure you definitely use that with the oil immersion lens. And again, when you put the microscope away, make sure that you turn the stage way down so it isn't anywhere near the objectives, and make sure that you have the light turned all the way off. As far as glasses go, you shouldn't have to wear glasses while you have the microscope on. You can if you want. The problem is the microscopes we have are not what are called long eye relief microscopes, so usually having your glasses on will get in the way. But as I said before, if your eyes are slightly different, if you use that technique I told you about adjusting the oculars, then you can compensate for any differences in your eyes. And the other thing to go along with that is, most of the time you're in lab, we're going to require you to wear safety glasses, but anytime we're using microscopes, that'll be the one time when we will relent. As long as you're using the microscope, then you can have your safety glasses off also. There are a few things you keep in mind about looking at specimens. It's always good to look at your slide ahead of time, see if it's really dirty. You might want to clean it. Most of the ones we have should be fine because they're going to come directly out of the box. It's always good to look at your slide and see if you can get an idea where the specimen on your slide actually is. So when you put it in the microscope, you can put that part of the slide right under the objectives and not have to hunt around too much. As I said before, lowest power objective, adjust the illumination with the condenser and the iris diaphragm as much as you can, and start at the lowest power and move progressively to the higher power. One is so you can make observations of these power and see what's different, and the other is, as I said before, it's much easier to do the focusing on the lower power. Again, if you need the oil lens, make sure you use the oil lens, but make sure you use the oil only on the oil lens. And in the case of the microscopes in the lab, the oil lens is the 100x objective, so only oil on that one and none of the other ones. And be really careful. If you get oil on the other ones, just make sure we clean it off. And I know it's number 7 here will sound, funny. And you say, oh, it'll never happen to me. But one of the most common problems you can't get an image to focus is that you turn the microscope slide upside down. And if you remember when I showed you the thing about the working distance of the lens, if you turn it upside down, the slide is too thick for the working distance of the lens, and so you will never actually be able to focus all the way through the slide onto the specimen. And that's definitely true of the high powers. It depends. Sometimes you can actually get, for instance, the 10x objective to focus because it has a working distance that's long enough to focus all the way through the slide. So if you find that you can get it focused on the lowest power, but then it won't focus on the higher power, check and make sure that your slide is right side up. Happens to the best of us. Even I occasionally turn it the wrong way. The good thing is most of the slides we have in the lab right now have one frosted end, so you should be able to tell which side is right side up relatively easily. So the other thing that we will have in lab this week is, and every week, we have cameras, and you can take pictures of everything you see in the microscope. That doesn't, however, mean that you don't still have to actually draw everything that it says draw. Well, keep in mind when it says observe, you need to draw it, and you can, if you want, take a picture just so you can have a better image to use in whatever report format we ask for that week. And if you're not quite sure how to use the camera, because we're using them in sort of simple and funky way, flag us down or find one of your fellow students who probably has already been in the lab before and might know how to do it. All right, so there's all the sort of historical background and things on how to use a microscope. Now the cool part, the things we get to actually look at this week in lab. All right, so the fun thing is you're going to observe and draw an amoeba. The thing to remember about the amoeba will be, one is most of the time we buy commercial amoeba cultures, so we don't have to keep them going ourselves. And in general, what you'll find is the amoeba are going to be on the very bottom of the jar. So if you want one, what you want to do is reach in with the plastic transfer pipette and suck up a little bit of liquid directly from the bottom of the jar and get a little bit of that, as I will say, the technical term for it is crap that's on the bottom. So you'll see that there's some stuff which is mostly food for the bacteria that the amoeba then eat on the bottom. If you suck up a little bit of that and stick on the bottom, you probably will get an amoeba. The other things to keep in mind when looking at the amoeba are keep your light level, in this case, as low as you can. Because if you think about it, if you have the light turned really up way high and you have the condenser well focused and everything, you essentially put that amoeba under what almost amounts to a laser. It's like standing on the sun for the amoeba. So if you leave them that way very long, what you will see are many exploding amoeba. Okay? So be careful with that. We'll have example ones up on the screen for you to look at, but if you're not careful, you will actually fry your amoeba and you'll see lots of exploding ones. And you'll know they're exploding because you'll see stuff streaming out of the inside of them. We're also going to observe and draw the division of yeast. So you're going to start off with some dehydrated yeast, put it in growth media, and then over time look and see if they actually start to grow by counting the buds on the side. And I've given you a picture in the handout of what budding will look like, and if you're not sure, we can go over it in class. Call us over. We'll do our best to sort of talk you through it. That's the one we're actually going to collect class data, so remember to answer the questions to collect class data. And as I said, for those of you who have had class before, we've collected class data using the clickers. Now we're going to collect the class data through the computer and use a Google Doc. So it'll be a little bit different, but hopefully easier system to use. We're actually going to be drawing euglena, and that one's kind of fun because they're very fast and they're going to whip around. So get an idea of how hard it is to look at live organisms under the microscope, and then we'll show you there's a stuff called protoslow that you can use, which actually is a really thick polymer that will slow them down. And then we do a couple of different kinds of staining things. So you're going to draw and stain potato cells and stain them for starch granules. So you get really cool, surreal sort of purple photos out of this if you want to take pictures. And lastly, you're going to observe, draw, and stain some squamous epithelial cells. And it's a big long word for saying we're going to scrape some cheek cells and put them on a slide. So as I said, you'll be able to take pictures of all these, but you still need to draw them. There are 5 cameras in the lab, but you can bring your own camera if you want to try it. Remember that there are videos available for how to do all of these things, and the pre-lab will steer you to the wiki, which will show you where they are, and there will also be links if you need to while you're in class. While you're right there, you can call them up in the computer and look at them right in class. There'll be a link in the procedure. So that's what we're going to do. This is just an idea of what you're going to look at. So here's euglena in the different parts. You see there are lots of little green chloroplasts in euglena, and this is what you're generally going to see when you look at the live ones. They're going to be really green, a little red eye spot, which is sometimes hard to see, kind of here in this one. And they have long flagella, and they'll be whipping back and forth pretty quick until you put them in protoslow. We also have some commercial slides of staining euglena, which are easy to look at, but not so cool because now they're purple and they don't move. So that's euglena. Also, the other things we're going to be looking at are amoeba, and so you'll be watching them stream pseudopods around the microscope slide or screen if you're looking at the TV, and you'll see all these cool little things just sort of streaming along. If, as I said, you had it under the light too long, you'll see one of the cell wall burst, and you'll see the stuff stream out of them. So that's when you'll know you've had them under the light a little bit too long. We're going to look at yeast, and you see that if you get the light adjusted just right, they kind of have a little bit of a greenish cast, but in general they're going to be really clear, so they're hard to look at. And when you look at them under low power, they're going to look like little grains of dust. So the reason we use the yeast is that if you can figure out how to learn how to focus a microscope on yeast, then everything else after that is easy. So once you get this down, you should have no problem with the microscope. And the things we're going to be looking at is, here's a yeast that doesn't have any budding. So you see that it's just a plain sphere. And what you're going to be counting are ones like this one that have this little tiny bud coming off the side. See, like all these ones. So we can talk about in lab what you think is budding and what not. Right? The potatoes, what you're going to be staining are all these amyloplasts that have starch or starch granules with iodine, so they're going to turn purple. So that's pretty cool. And then your own squamous cells. So what you'll mostly see is a blue background and a really dark blue nucleus. So those are the other things that you're going to observe. And so that's pretty much the way your lab's going to go. So make sure you listen to the other lecture that Allie's going to do so you can get an idea of generally how things are going to go. Otherwise, we'll see all of you guys next week, or this week if you're not listening to this until then, in Goddard. And remember, for any of you who haven't paid attention, we have moved to Goddard Hall. So the lab is at GH203, and we're no longer in Salisbury. So I think that's all I have for you guys. Clearly you can't ask me any questions, but you can bring any questions you have to lab. And Al and I will see you next week. And I leave you with one last philosophical question of an actual electron micrograph. So in case you want to know how many bacteria can dance on the head of the pin, here you can actually count them. So that's it. And I guess I'll see you guys next week in person. So if you don't know what I look like, you can actually figure it out. I'll see you then.